What we're looking at here is our liver model for microscope questions. So we will look at liver histology, but we also have this plaque here showing all of these microscopic structures, some of which we can't actually see on our slides. So overall, we have these repeating units, these hexagonal shaped cells. These are hepatocytes. So they're the basic unit of the liver. Hepato or hepat means liver. So that's a Latin term to keep in mind. We do not see liver lobules. So we are not on that scale. Instead, we're on the cellular scale. We can see our hepatic triad. So we're going to have lots of branches coming off of these three vessels here that form our hepatic triad. Artery is going to be color-coded red. We have our vein in blue, and then this green guy is going to be dealing with bile. So let's start with that artery. This is going to be our hepatic artery. Makes sense because of that stem, that Latin stem, hepatic. We have a portal vein, and we will talk about that portal system in more detail in a future video. And then we have in green our bile duct. And all of these are going to be branching off and perforating into the tissue here. Other things we can see are the central vein. So we have a central vein here, here, they're scattered all over, and that is going to be forming the center of our hepatic or liver lobule, which I will not ask you from this view. We also have these tiny, tiny vessels in green. You can think of these as the capillary equivalent for bile. All of these are branching off of that bile duct, and we call these bile canaliculi. And if that term sounds familiar to you, you are correct. We also saw canaliculi when we talked about bone, specifically compact bone. And it's the same concept here. We've got these tiny vessels interfacing with our hepatocytes to transport bile. In terms of what you should review for the lab exam, definitely want to know your unit of the liver, which is this hepatocyte. Be familiar with these vessels of the hepatic triad, so artery, vein, and bile duct. And I also want you to be able to identify these bile canaliculi on this model.